Today we will discuss about this calreticulin silent cells effect on these EMT related pathways. So far we have discussed that calreticulin silent cells affect EMT, but we have not precisely see the effect, how they will affect the pathways that are uh, changed or modulated by EMT. So and the paper is focusing on three different pathways that are affected by EMT, that is integrin related pathways, EGFR-ERK related pathways, and MAPK pathways. So their focus is on these three pathways. So what they are able to see, in order to check the effect for EMT, they use the EGFR-induced cells. So they check the effect whether these EGFR related pathways are affected or not. So they check the phosphorylation of this EGFR at three different locations, that is at 1173, 1068 and 845. And what they are able to see is that they are able to see that in the case of these calreticulin silent cells, the EGFR 1173 is does not increase even when the EGFR is induced, whereas all the other, that is EGFR 1068, we can see there is a increase in the even in the case of calreticulin knockout. And similarly for EGFR 845, we can see that even in the case of calreticulin not knockout, silenced calreticulin silent cells with EGF induced, they are able to see the upregulation in the PGFR. But in the for the case of 1173, the increase is not almost similar to the levels. The level of PGFR 173 is similar to the level of these calreticulin silent cells that are not EGF induced, that are not induced by EGF. So from that they come to a point, they are able to see that changes they are able to see in the 1173. Thereafter what they did is did the immunofluorescence assay to check this effect, and they are able to see that for EGFR 1173 is increased in the case of EGF induced conditions, and for the control conditions it is we do not see any signals, that is without EGF induced cells, and this is for the KPN2 cells. And for the case of ASPC1 also, we can see that in the case of this EGF induced conditions, we are able to see increase in the PGFR 1170, but for the control case we do not see any increase in the signals for the 1173. And these things later on they quantified in the panel A, where we can see that for the, and this panel A is first for this KPN2 cells, where we can see that for the EGFR the increase is statistically significant compared to the case where EGFR is not induced. And similarly for 106, but for the 1068 and 845, they are able to see increase, but the increase is not statistically significant. Thereafter for the ASPC1 cells also, they are able to see the same thing, that for PGFR 1173 there is they do not see an increase, but for the other cells they are able to see increase. But for the other phosphorylations of the EGFR conditions, they are able to see increase even in the calreticulin silence condition. So their focus is on the PGFR 1173 in ASPC1 cells also, and here also they are able to see in the 1173 conditions the increase is statistically significant, but for the case of 1068 and 845 the increase is not statistically significant. And the reason is that they are comparing the increase with respect to the scramble which are induced with EGF. So here the difference is statistically significant, but for the case of 1068 and 845 it is not. And similarly for the case of KPN2 also, compared to the scramble, do not see any statistical changes. And this immunofluorescence is to show that there is an increase in these 1173 signals in the EGF induced conditions for the KIP2 cells. So here the results are shown for the scramble transduced cells, not for the calreticulum silent cells. So that's all for this video. If you need to understand more about these experiments in detail, then you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.